Hi students, welcome to our zoology class, and today we are going to discuss a new experiment which is done by Frederick Griffith. Okay, a scientist named Frederick Griffith made a series of experiments in Streptococcus pneumoniae, that means the bacterium that causes pneumonia. So he made a series of experiments in that bacterium to find out some findings. Okay, let's go into that experiment in detail. When this Streptococcus pneumoniae, that is the bacterium that causes pneumonia, were cultured on a culture plate, some of it produces very smooth, shiny colonies. Okay, and that is termed as a strain of that bacterium, while others produce rough colonies. Okay, and that rough colonies is termed as R strain of that bacterium. So the smooth shiny colonies are termed as S strain and the rough colonies are termed as R strain. And this S strain has a polysaccharide coat over it, or it has a mucus cover or mucus layer over it, and they are pathogenic. That means they can cause disease. Which disease? Pneumonia. We know that Streptococcus pneumonia causes pneumonia on organisms. So they are the pathogenic. Which strain? S strain, which has a mucus or a polysaccharide coat, are pathogenic. That means they can cause the disease, what? Pneumonia. While the R strain, that means the rough colonies, they do not have any polysaccharide coat over it, and they are non-pathogenic. What do you mean by non-pathogenic? They can't cause any disease. That means they can't cause the pneumonia. This Frederick Griffith done his experiment on mice. Okay, and when he injected the mice with a strain of bacterium, that means they are virulent. That means they are pathogenic. And because of that, the mice have dead. Okay, while the R strain bacterium, while he injected the mice with R strain bacterium, the mice was not dead, because we already said that they are non-pathogenic forms. They can't cause the disease. Okay, so when Griffith injected the mice with S strain, the mice dead, while he injected the R strain, the mice was not dead. So here you can see the virulent bacteria as well as the avirulent bacteria. Virulent bacteria means the S strain, which means it causes, or when this bacteria injects over the mice will dead. Okay, avirulent bacteria means they are the R strain, rough colonies. They are non-pathogenic. So when they are injected to mice, the mice lives. So when the virulent bacteria or S strain was injected on mice, the mice dies of pneumonia, while the non-virulent bacteria is injected, mouse remains healthy. It won't be dead. Clear? That was actually the first step of Griffith experiment. Okay, and I hope you have cleared that portion. Okay, and on the second step, or in the second phase, he killed the bacterium by heating. Okay, while heating, the bacteria can be killed. Okay, so he, on the second step, he injected the mice with heat killed S strain. Okay, heat ചെയ്തിട്ടുള്ള S strain ആണ് മൈസിൽ ഇൻജെക്ട് ചെയ്തത് സെക്കൻഡ് സ്റ്റെപ്പിൽ. Okay, so normal case, we have already said that on the first case, or in the first step, when S strain were injected, the mice was dead. But when the heat killed S strains were injected, the mice was alive. It was not dead. Okay, and on the next part, he mixed the heat killed S strains and live R strain, because we have already said that R strain did not cause any disease on mice. So he mixed the heat killed S strains and live R strains on mice, but the result was amazing. That means that result in the mice was dead. Okay, the mice was not alive, it was dead. Okay, moreover, Griffith rediscovered the living S bacteria from the dead mice. Okay, നമ്മൾ മിക്സ് ചെയ്ത് ഇൻജെക്ട് ചെയ്തപ്പോൾ, that means living R strain heat killed S strain mix ചെയ്തിട്ട് നമ്മൾ മൈസിൽ ഇഞ്ചെക്ട് ചെയ്തപ്പം the mice was dead. Okay, and he rediscovered the traces of S bacteria, S living bacteria on that dead mice. Here you can see when this heat killed a strain of bacterium was injected, the mice was live. It doesn't cause the death of the mouse. Okay. So here in this image you can see when living R cells are injected, the mice was live, while living S cells were injected, the mice was dead. And when these S cells are heat killed, it doesn't cause the death. That means the mice was live. When the living R cells and heat killed S cells were mixed and injected, it also caused the death of the mouse. Clear ano? And based on this experiment, the Griffith concluded that the R strain undergoes some transformation or change by the presence of S strain or the heat killed S strain. Heat killed S strain in the presence, there undergoes some transformation to the R strain. Okay, and some transforming principle was transformed from the S strain, that means heat killed S strain, to the R strain, which enables the R strain to produce the polysaccharide coat over it and become virulent. We have already said that R strain do not have the polysaccharide coat, but when we mix that both the R strain and S strain, some transforming principle was transformed from this S strain to the R strain, and that transforming principle helped the R strain to produce a polysaccharide coat over it and become virulent or become pathogenic, and that's why that R strain caused the mice to death. Okay, this may happens due to the transfer of genetic material. Okay, but the biochemical nature of that genetic material was not revealed by this Griffith experiment. So from the Griffith experiment, it's clear that there is the presence of some transforming principle. Okay, and for the determination of its biochemical nature, certain scientists known as Avery, MacLeod, McCarthy, they all conducted some test or biochemical test in order to find out the biochemical nature of that transforming principle which is revealed by the Griffith experiment. And they purified the biochemicals. Biochemicals means DNA, RNA and protein from the heat killed S cells. നമ്മൾ ആ ഹീറ്റ് കിൽഡ് എസ് സെൽസിൽ നിന്നും അതിൻ്റെ DNA പാർട്ടും RNA പ്രോട്ടീൻ എന്നുള്ളതൊക്കെ പ്യൂരിഫൈ ചെയ്തു in order to find out which material transformed the live R cell into what? S strain. R strainനെ S strain ആക്കി മാറ്റിയത് ഇതിലെ ഏത് കമ്പോണൻ്റ് ആണ് എന്ന് മനസ്സിലാക്കാൻ വേണ്ടിയിട്ടാണ് they had purified the biochemicals from the heat killed S strains, and they found out that DNA alone was the reason for the transformation of this R strain into S strain. Okay, live R strainനെ virulent S strain ആക്കി മാറ്റിയത് DNA എന്നുള്ള ഒരു കമ്പോണൻ്റ് ആണ് എന്നുള്ളത് മനസ്സിലാക്കി. And they also discovered that proteases, that means protein digesting enzymes, and RNases, RNA digesting enzyme, do not affect the transformation. ഈ പറയുന്ന ലൈവ് ആർ സ്ട്രെയിനെ എസ് സ്ട്രെയിൻ ആക്കി മാറ്റുന്ന ആ പ്രോസസ്സിനെ ഒന്നും അഫക്റ്റ് ചെയ്തിട്ടുണ്ടായിരുന്നില്ല. ഏത്? പ്രോട്ടിയേസ് ആൻഡ് RNAs. That means that genetic material may not be protein or RNA. അങ്ങനെ കൺക്ലൂഡ് ചെയ്യാം. While at the same time the digestion with DNAs, okay, that means the enzyme that digests DNA had inhibit that transformation. Live R strainനെ S strain ആക്കി മാറ്റുന്ന ട്രാൻസ്ഫോർമേഷൻ പ്രോസസ്സിനെ ഇൻഹിബിറ്റ് ചെയ്തു. എന്ത്? DNAs. DNA digesting enzyme is known as DNAs. So this, when DNAs are given, it affected or it inhibited the process of transformation. That means the genetic material would be a DNA. ഓക്കെ, അപ്പോൾ പ്രോട്ടിയേസിനും അതേപോലെ RNAസിനും ഒന്നും ട്രാൻസ്ഫോർമേഷൻ പ്രോസസ്സിൽ യാതൊരു റോളും അല്ലെങ്കിൽ ഒരു ഇൻവോൾവ്മെൻറ്റും ഉണ്ടായിരുന്നില്ല. ദാറ്റ് മീൻസ് that genetic material may not be a protein or a RNA, while at the same time the DNAs, That means the enzyme that digests the DNA can inhibit the transformation, means that DNA would be the genetic material. From this discovery, the scientists concluded that DNA is the hereditary material. Okay, അപ്പോൾ നമ്മൾ DNA is എന്ന് പറയുന്ന എൻസൈം ട്രാൻസ്ഫോർമേഷൻ ഇൻഹിബിറ്റ് ചെയ്തപ്പോൾ they concluded that DNA would be the genetic material, അല്ലെങ്കിൽ DNA ആണ് ഹെറിഡിറ്ററി മെറ്റീരിയൽ എന്നുള്ള ഒരു കൺക്ലൂഷനിൽ ആ സയൻറ്റിസ്റ്റൊക്കെ എത്തി. ബട്ട് ഓൾ ബയോളജിസ്റ്റും അതിന് കൺവിൻസ്ഡ് ആയിരുന്നില്ല ഈ ഒരു എക്സ്പെരിമെൻറ്റിൽ. ബട്ട് സ്റ്റിൽ they have concluded that DNA would be the genetic material. So that is about this Griffith experiment. അപ്പോൾ ഈ ഒരു എക്സ്പെരിമെൻറ്റ് വഴി ഏകദേശം ജനറ്റിക് മെറ്റീരിയൽ DNA ആണ് എന്നുള്ള ഒരു ബേസിലോട്ട്, എന്നൊരു ഫൗണ്ടേഷനിലോട്ട് എത്തിയിട്ടുണ്ടായിരുന്നു. ഓക്കെ, I hope it is clear to you. So the next portion will be covered on the next class. That's all for today and thank you.